Good afternoon, dear colleagues. Today we will continue our series of our webinars. Today we will pay attention and we will discuss about classification of adverse reaction. Let me introduce myself for our new participants. I see and I'm very thankful that many questions I have received. I received this question, and I think that today I will reflect all needs concerning on classification of adverse reaction. About myself, I would like to tell several words. My name is Galina Cardera. I'm head of pharmacovigilance department, QPPV clinical expert and medical doctor with clinical experience working in hospital and practical healthcare professional activities. We have discussed it in our last webinars concerning on IB classification, pharmacological classification of adverse reaction. Today we will repeat it because repetition is matter of studiorum, and after it we will go ahead. IB classification based on perceived relation of the adverse effect of the drug to dose and mechanism. Why we will repeat it in our webinar? Because for refreshing knowledge and for refreshing terminology. And about augmented classification, augmented adverse reaction, its type A reaction are the result of an exaggerated but otherwise normal pharmacological action of a drug given in the usual therapeutic dose. They are usually dose dependent. Normal implies that an abnormal pharmacological effect might be observed nonsense, and usually some are not dose dependent. This question you should ask for yourself when you make assessment. Be bizarre. Type B reaction are totally aberrant effects that are not expected from the known pharmacological action of a drug when given in the usual therapeutic doses to patients whose body handles the drug in the normal way. What question you should to ask is what about dose dependency? And concerning classification, and it's C and D and E type of drug reaction. Short-term reaction is A and B reaction, long-term reaction is C, D, a delayed E, E, F. C, D, A, and F categories do not specify dose dependency. We will continue this topic in types of adverse reaction. Type I augmented, dose-dependent, predicted from the known pharmacology of the drug. As example, is hypoglycemia can be insulin. B bizarre, unpredictable, dose-independent, rare, fatal. It can be anaphylaxis as example for penicillin. C chronic, prolonged treatment, analgesic neuropathy. D delayed after years of treatment, antipsychotic turdai can be dyskinesia. E end of use, withdrawal effect, GC withdrawal, adrenocortical syndrome. Classes of dose-related adverse reaction we will talk later. Now we will stop it on several classes. There are 3 classes of dose-related adverse reaction which are classified by the concentration and with the occur relative to the therapeutic concentration in an individual as follows. It can be toxic reaction, adverse reactions that occur and a therapeutical concentration, collateral reaction, reactions that occur at standard therapeutic concentration, and hypersusceptibility reaction, reactions that occur at subtherapeutic doses in susceptible patients. Type A reaction as a mechanism is predicted from the known pharmacology of the drug. These reactions are dose dependent. Examples can be bleeding with anticoagulants too. Type B bizarre reactions are not predicted from the known pharmacology of the drug. They appear but actually are not relatively dose independent, as very small doses might already elicit symptoms. They include immune-mediated side effects like maculopapular exanthema, but also other hypersensitivity reactions like aspirin-induced asthma. Type C chemical chronic, which are related to the chemical structure and its metabolism, for example, paracetamol hepatotoxicity. It's now a very frequent symptom, especially in West European country, because this pandemic, COVID pandemic and influenza increases administration of this drug, and it can be very careful about syndrome Ray syndrome is about paracetamol hepatotoxicity. I am preparing, and it will be another webinar concerning on drug-induced hepatic disorder, hepatic disease, and we will talk about all symptoms, all cases concerning on hepatotoxicity. Type D, delayed type of reaction. This type appear after many years. It's prolonged treatment. For example, bladder carcinoma after treatment with cyclophosphamide. Type E, end of treatment, occur after drug withdrawal, seizures after stopping, for example, phenytoin. Type A, augmented, is assumed type reaction result from an exaggeration of a drug's normal pharmacological action when given in the usual therapeutic dose and are normally dose dependent. Examples include respiratory depression with opioids or bleeding with warfarin. Type A reaction also include those that are not directly related to the desired pharmacological action of the drug, for example, dry mouth that is associated with tricyclic antidepressant. Type A, augmented type reaction which can be predicted from the known pharmacology of the drug. Again, it's dose-dependent type of reaction, can be elevated but a dose reduction. For example, anticoagulants bleeding, beta blockers bradycardia, nitrates headache, and prazosin postural hypotension. Type B, bizarre reaction, cannot be predicted from the pharmacology of the drug. They are less common and so may only be discovered for the first time after drug has already been made available for general use. Not dose dependent. Host dependent factors important in predisposition. For example, it can be penicillin anaphylaxis, anticonvulsant, hypertensivity. Examples of bizarre reaction can be hypersensitivity reaction, Stevens-Jones syndrome, or hemolytic anemia. Type B reaction are novel responses that are not expected from the known pharmacological action of the drug. They are less common and so may only be discovered from the first time after a drug has already been made available for general use. Examples include anaphylaxis with penicillin or skin rashes with antibiotic. Type B reactions are defined as a totally aberrant effects that are not be expected from the known pharmacological action of a drug when given in the usual therapeutic doses. If all adverse effects of drugs are dose-related, the labeling of some adverse reaction as non-dose dependent is misleading, as this definition implies that it is dose for concentration at which an adverse effect occurs that defines it to be or AZ type or A or B. Note the dose relatedness of the effect by the position of the dose response curve. However, the concept of a usual therapeutic dose is also flawed for several reasons. There is often wide variability in responsiveness to a given dose of a drug. For some drugs, for example, dosage vary according to indication. For example, the effective dosage of vitamin D in dietary deficiency is 10 micrograms per day compared with 2,500 micrograms day in renal osteodystrophy. The effective dosage of clonidine is preventing migraine may be as low as 100 micrograms per day compared with up to 1,200 micrograms day in hypertension. The usual dosage of bronchyptine in cyclin B in breast disease is about 5 milligram per day compared with up to 40 milligram per day in Parkinson's disease. Dose responsiveness can change from time to time, even in the same individual. For example, if there is tolerance as with opioids. Example can be erythromycin, dose-dependent nausea and vomiting. It could be classified as A, dose-dependent. It could be classified as B, it is not predictable effect from its antibacterial normal action. And what about adverse reaction, which onset depends on the duration of therapy? And type C and E. Type C continuing reaction describes those that persist for a relatively long time, for example, osteonecrosis of the jaw with bisphosphonates. Type D delayed reaction which become apparent sometime after the use of medicine, for example, leukopenia, which can occur up for 6 weeks after the dose of lomustine. Type E end-of-use reaction are associated with the withdrawal of a medicine, for example, insomnia, anxiety, and perceptual disturbance following withdrawal of benzodiazepines. Short-term reaction A and B, long-term reaction C and D, a delayed reaction E and F. CDA category do not again specify dose dependency. Reaction type C, more precisely, its chemical reaction, its biological characteristic can be predicted from the chemical structure of the drug metabolite. Again, paracetamol hepatotoxicity. Long-term effects are usually related to the dose and duration of treatment. Examples: ethambutol, butol retinopathy, nesteroid NSAIDs, nephrotoxicity. Type D delayed reaction occur after many years of treatment, can be due accumulation, chemotherapy, secondary tumors, phenytoin during pregnancy, for example, teratogenic effects, antipsychotic, tardive dyskinesia, analgesics can be cause of nitrotherapy. Type E end of treatment reaction occur on withdrawal, especially when drug is stopped abruptly. For example, phenytoin withdrawal seizures, steroid withdrawal adrenocortical insufficiency, withdrawal syndromes. Examples: benzodiazepine rebound insomnia, agitation, clonidine rebound hypertension, corticosteroids acute adrenal insufficiency. Drug withdrawal reaction, sudden interruption of drug, worsen clinical condition for which previously drug was used. Examples: a corticosteroids in asthma can be cause of precipitate acute attacks, myalgia, depression. Beta blockers, worsening of angina, precipitates myocardial infarction. Now we will go to new classification. It's EIDOS and DOTC classification. EIDOS describes aspects of the mechanism by which the adverse effects arises. DOTS describes the clinical features of the adverse effects once it has occurred. Together, there are two classification systems comprehensively delineate in the important aspects of adverse reaction, taking into account the extrinsic medical species that initiated the effect, in case of its metabolites, the intrinsic chemical species that it affects, the distribution in the body of these species, the physiological or pathological outcomes, the adverse effect, and the sequelae adverse reaction can be classified using DOTS. This classification can be considered as mechanistic. Comparing on with clinical features of adverse reaction, it's DOTS. Classification, it's another one where you should take into account dose, timing, in as susceptibility of and these two classifications we will discuss in this webinar. EIDOS DOTS classification. EIDOS again describes aspects of the mechanism by which the adverse effect arises. DOTS describes the clinical features of the adverse effect in for example, the adverse reaction once it has been occurred. We will now stop it the EIDOS mechanistic classification of adverse drug effects. We will discuss now about features, varieties, and examples. Reflect it. For example, extrinsic species can be, and we see it as a varieties, it can be parent compounds. As example can be insulin, an excipient, polyoxyl certified castor oil, a contaminant, for example 1.1-ethyl adenylbase tryptophan, an adulterant, lead in herbal medicines, a degradation product formed before the drug enters the body, outdated tetracycline, a derivative of any these, for example, a metabolite can be acrolein from cyclophosphamide. When we are talking about intrinsic species and the nature of its interaction, we will stop the extrinsic species. For example, first one is molecule. It can be at varieties, nucleic acids, DNA, RNA. DNA, it can be melphalan. At the RNA, it will be mitoxantron. Enzymes, reversible effects, as example can be adrenophonium, and irreversible effects can be mollation. And receptor, reversible effect can be prostacyclin, and irreversible effect can be phenoxybenzamine. Ion channels transport as calcium channel antagonism, digoxin, for example, natrium, calcium ATFase. Other proteins, immunological proteins, penicillyl residue hapten, tissue proteins, and acetyl p-benzoquinolamine, paracetamol, for example, as acetaminophen. Another one is the intrinsic species and the nature with interaction with the extrinsic species is extracellular. It can be water, dextrose as example 5%, hydrogen ions, pH, sodium bicarbonate, and other ions, potassium concrenoate. physical or physicochemical, direct tissue damage in the intrathecal vincristine, altered physiochemical nature of the extrinsic species, Sulin-Duck precipitation. Distribution. Where is the body the extrinsic and intrinsic species occurred, affected by pharmacokinetics. For example, its antihistamines cause drowsiness only if they affect histamine H1 receptors in the brain. Outcome, the physiological or pathological change, the adverse effects, dose, time, toxicity, and sequel, the adverse reaction. Examples of physiological and pathological changes in adverse drug reactions, some categories can be broken down further. We will now consider and take into account the type of change, and we will now discuss examples too. Physiological changes can be increased actions. It can be hypertension, monoamine oxidase inhibitors, clotting, tranexamic acid, decreased action, bradycardia, beta-adrenoreceptors antagonists, QT interval prolongation, anti-arrhythmic drugs. Cellular adaptation, lipoatrophy, subcutaneous insulin, glucocorticosteroid-induced myopathy. Atrophy. It can be induced myopathy, hypertrophy, gynecomastia, spironolactone, and hyperplasia, pulmonary fibrosis, busulfan can be cause of it, and retroperitoneal fibrosis, methylsergide. Type of change is metaplasia. Examples: lacrimal canalicular squamous metaplasia. It can be caused medicine fluorouracil. Neoplasia, hepatoma, anabolic steroids. It can be benign, for example, in malignant. In own cases, can be hormonal pathogenesis and cause to vaginal adenocarcinoma, medicine diethylstilbestrol, genotoxin, transcendental cell carcinoma of bladder, cyclophosphamide, and immune suppression, lymphoproliferative tumors, cyclospore, altered cell function, LGA-mediated mast cell degranulation, class 1 immunological reaction. Cell damage. Acute reversible damage can be chemical damage, as example periodontitis, local application of methylene dexamethamphetamine, immunological reaction, class 3 immunological reaction. Irreversible injury, cell lysis, class 3 immunological reaction, necrosis, class 4 immunological reaction, hepatotoxicity, paracetamol after apoptosis, and precisely apoptosis, liver damage can be cause of this reaction, troglytazone. Intracellular accumulation, milk alkali syndrome, calcification, crystal storing histiocytosis. It can be clofazimine, skin pigmentation, amiodarone, and drug deposition, skin pigmentation. Extracellular accumulation, urinary calculi, it can be triamterene. When we will compare this classification, you see that AB to compared EIDOS plus DOTC, we will have full picture, and we will now to see what types of drug reaction can happen and how classificate it, taking into account some definite factors. AB classification is defined only by properties of the drug. It can be pharmacology and dose dependency. EIDOS plus DOTC is comprehensive classification, including properties of the reaction, time course of the reaction, intensity, and property of the patient, genus, underlying pathologies, biological differences, then confer susceptibility. How the question to be asked at each stage of the EIDOS classification process and those relationship, time course, and susceptibility. For example, when we asking about nature of the extinct species, you should ask what's the nature of the extinct species and SI was the nature of the intrinsic species. As it go to distribution, is E distributing to the same side as I, and E plus I going to distribution, was the nature of the interaction between E and E1 and I. It's molecular, it can be what nature, extracellular, psychochemical. What is the physiological, pathological outcome, and what's the sequela, what's the DOTS classification? When assume it, the DOTS classification is a clinical classification and observed features of adverse drug reaction. It could be used, for example, in pharmacovigilance planning and developing regulatory strategies for dealing with new adverse drug reaction after a drug has begun marketing authorization. EIDOS, which considers the mechanism of adverse drug effects, extends the idea of rational classification to encompass non-clinical information. When relevant data are absent, EIDOS provides a framework for assembling them. It cannot, of course, give decision in the absence of such data. Extrinsic species in medicines. It includes drug molecule itself, an excipient, an contaminant, an adulterant, a degradation product, and derivative. Identify these extrinsic species that cause adverse reaction has important practical consequences. For example, breakdown product of tetracycline cause Fanconi syndrome. This causes the manufacturers to decrease the shelf life of tetracycline. The extrinsic species involved in adverse reactions. The drug molecule itself, for example, indomethacin, inhibits renal prostaglandin synthesis and can cause renal impairment as a result. An excipient, for example, polyoxyl certified castor oil, crema 4L, which is used to make aqueous solution of lipid-soluble drugs, such, for example, as cyclosporine, sandimunor, can cause non-H-mediated anaphylactic reaction. A contaminant such as 1,1-ethyl-dibenzyl tryptophan, L-tryptophan, a byproduct of manufacture represent certain batches produced as anaphylia myalgia syndrome in patients treated with tryptophan. An adulterant such as lead or arsenic in herbal remedies or oversulfated chondroitin sulfate deliberately introduced into batches of heparin. A degradation product before introduction into the body, such as the degradation products in outdated tetracycline that have led to renal tubular damage. A derivative of one of the above, particularly metabolite of the parent compounds, for example, the hepatoxin hydrolysine, a metabolite of isoniazid. Identify the type of extrinsic species responsible for an adverse effect. Identifying the type of extrinsic species responsible for an adverse effect has practical implication. If it's the parent drug, the drug may have to be avoided or its dosage reduced. An excipient, contaminant, or adulterant can be removed from the formulation. When breakdown products of tetracycline were identified as a cause in Fanconi syndrome, the limited shelf life of tetracycline was recognized. The identification of acrolein as a metabolite of cyclophosphamide led to the introduction of methanol to prevent hemorrhagic cystitis. The intrinsic species and the form of interaction with the extrinsic species. There are three types of interaction between extrinsic chemical species and an intrinsic chemical species. For example, a tissue or fluid in the body then can result in adverse effect. Molecular interaction. Most interactions between the extrinsic species and the intrinsic species are molecular, will bind with some affinity to one or more intrinsic molecules. Alteration in the extracellular environment. Some adverse effects result from alteration in the composition of the extracellular fluid, for example, by dilution, alteration of hydrogen ions, or alterations in the concentration of solutes, such as sodium or potassium ions. Physical or physicochemical effects of adverse effects sometimes result from physical or physicochemical effects such as direct tissue damage or precipitation of a drug, for example with the renal tubulus, in the bile, or in the stomach. Molecular interaction. The intrinsic molecules that can be involved in interaction with extrinsic molecules include nucleic acid covalent adduct between cellular and DNA and alkylated agents such as melphalan probably explains the increased risk of leukemia in patients treated with these drugs. Enzymes. Most drug action involves enzymes and via enzyme inhibition. Some drug interaction involve induction. It can be reversible effects, as for example the peptidase that metabolize angiotensin 1 to angiotensin 2, angiotensin converting enzyme ICA also metabolizes bradykinin. ICA inhibitors therefore inhibit the breakdown of bradykinin. Fourth, increased concentration probably explains the adverse reaction of angioedema and Koch. Drugs that inhibit or induce enzymes related to drug metabolism are also associated with adverse effects. So, drug interaction, and these are included as accessibility factors in the dose classification. Inversible inhibition affects inhibition of platelet oxygenase of aspirin, acetylsalicylic acid, with a consequent tendency to hemorrhage that persists for several days after the end of treatment is irreversible. Receptor. As a molecular interaction, the intrinsic molecules, pharmacological receptors, including, for example, neurotransmitter hormone and cytokine receptors, are the main targets for the action of drugs. Adverse effects can result from altered receptor action in the target organ or elsewhere, either by a direct effect on the drug or on the receptor, for example, agonist and antagonist, or by indirect effects, for example, neurotransmitter releasers or inhibitors of neurotransmitter reuptake of metabolism. For example, phenphylamine increases the release of serotonin, which stimulates the serotonin 5-HT2B receptor, activating kinase that stimulated cell growth in pulmonary valve tissue, causing pulmonary valvulation. Irreversible receptor inactivation by covalent interaction, for example, inhibition of a A adrenoreceptors by phenoxybenzamine can lead to collateral adverse reaction. Ion channel transporters, inhibition of potassium channels in the digital convoluted tubula of the kidney by triamterene causes hyperkalemia. Excess inhibition of the natrium plus kalium plus 2-chlor-CO transporter by loop diuretics in the loop of the handle in the kidney causes hyponatremia and dehydration. Other proteins, immunological effect of the four types classified by Cell-Incombs can result from interaction between large extrinsic molecules, for example peptides and proteins, and intrinsic proteins of cells, from direct effects or components of the mute parties, or by formation of immunogenic ducts, as exemplified by the haptens formed between the penicilloyl moiety of beta-lactam antibacterial agents and intrinsic proteins. Such effects include the interaction between penicillin and IgA, causing anaphylaxis in sensitized individuals, or between proteins in horse serum and IgG, leading to serum sickness in those treated with horse-derived snake antivenoms. Tissue protein direct damage to structure proteins can cause functional derangement, and happens when the metabolite N-acetyl p-benzylquinoamine binds covalently to sulfhydryl containing hepatic and renal proteins after paracetamol acetaminophen poisoning. Alteration of the extracellular environment. Alteration of extracellular environment, the extracellular environment can change as a consequence of molecular interaction of the types mentioned above, as happens, for example, when chloropropamine acts on vasopressin receptors. However, there can be direct or non-specific effects of the components of the extracellular environment. This can affect water, hydrogen ions, pH, and other ions. Water crystalloids, such as 5% dextrose, can cause water intoxication directly. Absorption of cyclin solution during transurethral resection of the prostate gland can have important, albeit transient, effect of cardiovascular and neurological function. pH changes in hydrogen ion concentrations can lead to important local or systemic adverse effects. Large volume infusion of solution, such as sodium chloride 0 or 9%, that contains strong anions, can cause metabolic acidosis. Other ions, higher concentration of caution, such as sodium, can result directly from the use of antibacterial drugs made up as salts, such as sodium ticarcillin. Physical or physical-chemical interaction. Physical or physical-chemical interaction, examples of adverse reactions that result from physical-chemical effects include renal calculi due to precipitation of triamterene in the renal tract, and gallstones due to precipitation of sulindac in the biliary tract. Corrosives, for example, phenol used for nerve ablation, can cause nonspecific tissue damage. Distribution. The extrinsic and intrinsic species which interact only when they are both found in the same planes. The pharmacokinetics of the extrinsic species can affect the occurrence of adverse effects. For example, histamine H1 receptor antagonist antihistamines, such as chlorpheniramine, that cross the blood-brain barrier, can act on CNS histamine receptors and cause drowsiness. Never antihistamines such as cetirizine do not generally cross the blood-brain barrier in significant amounts and do not reach SNSH1 receptors, they therefore do not cause drowsiness. Outcomes. Outcomes of the interaction, pathological adverse effects, interaction between extrinsic and intrinsic species in the production of adverse effect can result in physiological or pathological changes. Some adverse effects arise through a combination of mechanisms. Sequelae. Adverse reaction. Sequelae, adverse reaction of the pathological changes induced by a drug continuity, the final step in the EIDOS classification, and describes the clinical recognizable adverse drug reaction. There may be more than one sequela of adverse drug effect. Sequelae can be classified using DOT system, those producing as a combined mechanistic and clinical pharmacological classification. Those a clinical pharmacological classification define the AB classification we talked about is more dose classification based on dose relatedness, timing, and patient susceptibility takes those factors into account. Those approaches to classify adverse reaction as summary. DOTS is developed by Aronson and Fernell. This approach describes properties of an individual adverse reaction such as dose, time, and susceptibility. Those can be divided into subtherapeutic, therapeutic, and supratherapeutic. Reactions that also occur at subtherapeutic dose are called hypersensitivity, which can be allergic and non-allergic in nature. Collateral effects are reactions that occur at therapeutic doses, similar to time reaction at normal dose. Reactions at subtherapeutic dosage are toxic reaction or exaggerated pharmacological effects in higher dosage of toxic drug levels. Timing. Some adverse reactions follow specific time patterns, others are time independent. Time dependent reactions are subdivided in rapid reaction related to high infusion rates, first dose, allergy or collateral, like hypertension with ICE inhibitors, early reaction, days to weeks, allergy or collateral, such as nitrate-induced headache. Some are persistent, others are self-limited and disappear while continuing the drug. Intermediate, weeks or 2 months, and late after chronic uses cumulative dose, and delayed, slow development of the adverse reaction. Time-independent reaction can occur at any time during therapy because of their nature or due to altered patient-related aspects like changes in kinetic or exogenous effects of drug interaction. Susceptibility. General risk factors for pharmacological adverse reaction are altered pharmacokinetics, age, renal function, altered metabolism. Risk factors for hypersensitivity reaction are ethnicity, genetic, previous sensitization. For some adverse reactions, specific risk factors are known in literature. DOTS is a clinical pharmacology classification. Again, system based on dome relatedness, timing, and patient susceptibility take those factors into account. Classes of dose-related adverse reaction. There are 3 classes of dose-related adverse reaction which are classified by the concentration at which they occur relative to the therapeutic concentration in an individual as follows. It can be toxic reaction, adverse reactions that occur as supratherapeutical concentration, collateral reaction, reactions that occur as standard therapeutic concentration, and hypersusceptibility reaction, reactions that occur as susceptibility doses in susceptible patients. Toxic reaction. Toxic reaction, a toxic is one that occurs as an exaggeration in the desired therapeutic effect. For example, syncope due to nitrate is a toxic reaction. It occurs by the same mechanism as a therapeutic effect, vasodilatation. Collateral reaction. Collateral reaction generally occur in a tissue other than in which the therapeutic action is sought, although not necessary in another organ. They can occur either through the same pharmacological effect as they vary by the therapeutic action is produced, for example, color, vision, disturbance from sildenafil, or through a distinct pharmacological effect, for example, dry mouth, due an anticholinergic effect of tricyclic antidepressant. Collateral effects are the most common cause of adverse reaction in this classification. The term collateral reaction has been coined for adverse reactions that occur at standard therapeutic concentration because the term side effects is often colloquially used to refer to all adverse reactions. Furthermore, VCO definition, World Health Organization definition, says ambiguously that a side effect is related to the pharmacological properties of the drug. In addition, side effects can be beneficial, as some authors have defined a side effect as an intended effect of medicinal therapy, whether good or bad. Because of the potential confusion, it may be best not to use the term side effect at all. The term adverse effect on reaction encompasses all types of deleterious effects. It makes no assumption about mechanism, evokes no ambiguity, and avoids the risk of misclassification. Hypersusceptibility reaction can be immune-related or not, and the term hypersusceptibility evokes implication about whether the mechanism is immunological on hypertensivity and on susceptibility of adverse reaction. The course of adverse drug reaction, time-independent reaction. There are two categories of time-relatedness of adverse reaction: time-independent and time-dependent. Time-independent reaction, time-independent, occur at one time during a course of therapy independent of duration of the course. They typically occur when the amount of drug being administered changes by the pharmaceutical mechanism, for example, by altered availability from a pharmaceutical formulation. When the concentration of the drug at the site of action changes by a pharmacokinetic mechanism, for example, dioxin toxicity when renal function worsens. When the pharmacological response is altered by a pharmacodynamic mechanism without a change in concentration at the site of action, for example, dioxin toxicity in association with potassium depletion. When such a reaction occurs, the delay before it occurs may be affected by the pharmacokinetic of the drug, but they are not as aspect of the time dependency as defined here. Time-dependent reactions are of 6 subtypes: rapid or immediate, first dose, early, intermediate, late, and delayed. Time-dependent reaction, rapid or immediate reaction, occur when a drug administered too rapidly, for example, the red man syndrome with vancomycin. They are typically toxic reaction. First dose reaction occur after the first dose of a course of treatment and not necessarily thereafter. There are typically hypersusceptibility reaction, but not necessarily immunologically mediated. Examples include hypotension after the first dose or an AICE inhibitor and type 1 allergic reaction. In the later, the reaction is observed after the first dose of a course, whether or not there has been a record of previous exposure. 30% of those who develop anaphylaxis with penicillin have no such record. A previous sensitized exposure can be regarded as a cause of a change in susceptibility. Early reaction. Early reaction occur early in treatment when either abate with continuous treatment, early tolerant, or persist, early persistent. They are typically due to collateral effects. Early tolerant reaction are reactions to which patients develop tolerance. For example, nitrate-induced or nifedipine-induced headache. Early persistent reactions occur early and do not abate with time, for example, corticosteroid-induced hyperglycemia or nifedipine-induced peripheral edema. Intermediate reaction. Intermediate reaction is the most common type of adverse reaction in the classification. They occur after some delay, but during longer-term therapy, the risk falls. After a certain time, there is no reaction. There is little or no risk that it will occur later. This reaction can be collateral or susceptibility reaction. Examples are allergic reaction of type 2, for example, thrombocytopenia due to quinine. Type 3, for example, interstitial nephritis with penicillins. And type 4, for example, cutaneous allergy due to antihistamines. And the ampicillin, amoxicillin, pseudoallergic rash. Non-allergic reaction of this type includes increased risk of neutropenia with carbimazole and of venous thromboembolism with antipsychotic drugs. Intermediate reaction occur in population of individuals with different susceptibilities. Those at higher risk have the reaction and stop taking the drug. Those at low risk do not have the reaction can be regarded as a healthy survivor, so the population risk appears to fall with time. Late reaction. Late reaction including withdrawal reaction. Late reaction occur primarily or not all at first, but the risk increases with continued or repeated exposure. There are typically collateral effects. Examples include tardive dyskinesia with dopamine receptor antagonism and some adverse reactions to glucocorticoids such as osteoporosis. Withdrawal reactions are late reactions and occur when after prolonged treatment a drug is withdrawn or its effective dose is reduced. They include opiates and benzodiazepine withdrawal syndromes, hypertension after withdrawal, or clonidine or methyldopa, and acute myocardial infarction and after beta blocker withdrawal. Delayed reaction. Delayed reaction observed some time after exposure, even if the drug is withdrawn before the reaction appears. They are typically collateral reaction. Examples are carcinogenesis, for example, vaginal adenocarcinoma in women whose mothers took diethylstilbestrol during pregnancy, and teratogenesis, for example, phocomelia due to thalidomide. How to apply EIDOS and DOTS? For example, adverse reaction, pathological gambling due dopamine receptor agonism, extrinsic modesty, dopamine agonist, its pramipexole, intrinsic modesty, dopamine receptors D1, D2, distribution, brain, outcome, altered cell function, nature unknown, sequela, pathological gambling. For example, due to DOTS classification, pathological gambling due to dopamine receptor agonism, dose relation, collateral, time course intermediate, genetic D1 susceptibility, genetic D1 receptor allele D1 800TC, age younger 107 Parkinson, sex male, exogenous combined therapy with levodopa. Complementary to EIDOS and DOTS when we can compare extrinsic species, drug molecule, excipient, contaminant, adulterant, degradation program, a dose relation, benefit, harm. When we see this triangle, the complementary of the EIDOS and DOTS classification system, an adverse reaction occurs when medicine or product is administration to a patient. Adverse rare mechanistical EIDOS, a bold Roman text, by nothing that the extrinsic drugs. Patients were co-administration distributed with one or more intrinsic patients. Patients has pharmacological or other effects the outcome. producing the adverse effects, the sequelae. It can be further classified using DOTS clinical classification, italicizing, which defines the important aspect of the drug, the relation between the dose response curves for benefits and harm, the patient susceptibility factors, and the reaction its time course, reproducing from the its link reproducing from Ferner Aronson, EIDOS 2010. To complementary form of classification of adverse drug reaction adverse reaction occurs when medicinal product is administered to a patient, read uppercase text. Adverse reaction can be classified mechanistically, EIDOS, blue italicized text, by or more instreaks species, has a pharmacological or other effects the outcome, producing the adverse effects the sequela. It can be first classified using DOS, the clinical classification, green board in text, which defines the important aspect of the drug relationship between the dose response curves for benefits and harm, the patient susceptibility factors, and the reaction its time course. Case study. 55-year-old woman admitted to hospital with shortness of breath, cough, and fever. EKG showed atrial fibrillation with rapid ventricular rate and normal QT interval. Electrolytes within normal range. Diagnosis: pneumonia. Treatments: erythromycin. Response: premature ventricle contraction after the first dose erythromycin. After 30 minutes of the second dose of the patient experienced torsade de points. Again, after 30 minutes of the third and fourth dose, torsade de points was noticed. The treatment was discontinued, a pacemaker was inserted. This potentially fatal complication of commonly used antibiotic is rare. Increased physician awareness is important, especially in patients with predisposing factors like electrolytes abnormalities, use of class 1A and class 3 antiarrhythmic drugs, and presence of prolonged QT interval, congenital prolonged QT syndromes. Torsade de points of erythromycin, it can be polymorphous ventricular tachycardia, examples of design and medical event, as examples are Stephen-Jones syndrome, anaphylaxis, aplastic anemia. QRS amplitude varies with characteristic illusion of the twisting of the QRS complex around the isoelectric baseline. Torsade de point is associated with a prolonged QT interval. Torsade is a life-threatening arrhythmia and may present a sudden cardiac death in patients with structurally normal hearts. Macrolide antibiotic, bacteriostatic, bacterial and higher doses, active against aerobic gram-coccus bacilli, usually prescribed for pulmonary infection and to patients that are allergic to penicillin. The association of erythromycin with torsade de points has been reported in 18 cases to date. Erythromycin has been shown to produce electrophysiological effects similar to those of class 1 and class 3 antiarrhythmic drugs or the cardiac muscle. At low doses it used as prokinetic. Patients prone to electrolyte abnormalities, hypokalemia, hypomagnesemia, hypocalcemia should be checked frequently to avoid lethal tachyarrhythmia. Whenever possible, rapid intravenous infusion of erythromycin which achieve large serum concentration should be avoided. Erythromycin, why can be happen? Erythromycin prolongs all phases of the action potential. The ability to delay repolarization could explain its tendency to produce prolongation of the QT interval. Action potential, no effect at 10 mg per liter, moderate change at 50 mg liter, a substantial alteration 100 mg liter. Factors that can alter erythromycin metabolism can enhance plasma concentration, hepatic damage, co-treatment with a drug that inhibits cytochrome 450, grapefruit juice. The definition of drug class effect. we can see that every class of drug has specific drug class effect. The definition of drug class effect is based on three concepts: a similar chemical structure, for example, the dihydropyridine ring of some calcium channel blockers, a similar mechanism of action, beta blockers block adrenaline receptors, and similar pharmacological effects, antihypertensive, anti-anginal, etc. Methods of detection adverse reaction. Hypothesis generated methods include spontaneous adverse reporting, with the systems whereby and suspected adverse reaction are voluntarily notified by health professionals, pharmaceutical companies, and other stakeholders to a central authority, Central Drug Standard Control Organization, for example, CDCO in India, or for example, PRAC now in Europe, or for example, regulatory authority in every country who control efficacy and safety of drugs. Prescription event monitoring represents a method which is a hybrid of spontaneous reporting with aspects of formal epidemiological studies. Systematic methods, public health surveillance data, such as death registers, are used to identify patterns of reactions that might be associated with drug use. Flexibility to adverse drug reactions. The risk of adverse reaction differs among different members on exposed population, and this is partly determined by different susceptibility. Susceptibility in this context is the capacity to have an adverse reaction. Susceptibility reactions are adverse reactions that occur in subtherapeutic doses in susceptible patients. Hypersusceptibility can be due to genetic factors, age, sex, physiological factors, for example, pregnancy, endogenous factors, for example, other drugs, foods, or disease, comorbidities such as renal insufficiency. Methods of detection adverse reaction can be hypothesis testing methods. It include case control studies. In case control studies, the research compares the exposure rate in the cases with the exposure rate in the control. Cohort studies. The studies involved a group of patients cohort followed up for a duration long enough to detect the outcome of interest. Randomized control trials. The studies involve patients divided into two groups randomly, into exposed and other not exposed, so that the outcomes can be compared. Four categories of safety respect of the amount of knowledge available. Well-established drugs. Drugs have been widely used for many 200 plus years, for which unlikely that completely identify safety usage will emergency. Established drugs for which there is substantial body of evidence of safety in clinical use but not enough to meet level 1. Provisional, newly authorized drugs until they have been used very extensively in ordinary practice over a period of at least 5 years. During this period, such drugs are normally under additional monitoring. Their safety in ordinary practice need to be studied proactively. Limited, all investigational drugs and the following situation where the drug might be authorized of limit safety information. Small population eligible for treatment, for example, rare disease treatment for which are known as orphan drugs. An example is culture disease, lysosomal storage disorder, which is usually treated with recombinant glucocerebrosidase. Drugs with important benefits of where there are in great clinical needs situation where potentially large risk might be acceptable, such as advanced cancer. Thank you for your attention. We today finished classification of drug reaction part 2, and I will be thankful if you will provide me your needs, your questions, your comments in my YouTube channel. Subscribe on my YouTube channels, and I will be useful in your practical activities. Concerning on future topics, it will be interesting topics in clinical pharmacology, and now I'm in stage of preparation and regulatory, and I'll be very thankful if you will provide topics which you want for providing a live webinars as workshop or a live seminars where you can ask questions, or for example we can thinking about round table where we can discuss normally what situation you have, how for example you make risk minimization measure, how it's working, how you control efficacy, because as we see now really that PRA committee now have a meeting every month, and we see how it's moving ahead. Plus MHRA now publicate new risk and for drugs, and there are some interesting journals. For example, I can advise you drug safety journal of Elsevier. It's nice one where you can find new articles. Another one, and it can be useful. Why I start talk about clinical pharmacology now? Because what I see is that many topics now medical doctor needs refresh knowledge, and hepatitis this subject is absent in universities as a pharmacovigilance, and this I am saying white spot can make dark life for medical doctor, because for example when I now will prepare medication errors, we can meet cases with death, hepatitis that doctors made medical errors, medication for example prescribing or different one. Concerning on question, we will go in specific question. It's pharmacovigilance for pediatric population. It's like announcement for future for elderly, and for example very interesting now materials and now in stage of preparation concerning on adverse reaction and vaccine, safety of vaccines and COVID vaccines, new dates, new analysis, and how work with it. Because from one side COVID opened box of Pandora, because we see that delayed adverse reaction can happen after, for example, 1 year, and different difficult to divide this effect from vaccine, or it can be effect of COVID, long COVID, how it's called now, or for example myocarditis, pericarditis in kids after vaccines, how work with it, how avoid. Another topic what I think will be very useful for you, you can ask question or for example give you a point of view concerning on medical topics, because what I see now it's what kind of situation you meet in real life as a medical doctor, what kind of situation do you really meet as pharmacist, what really situation you meet in daily activity. It's very important, and I hope and I pray that we will help for each other, and these lectures will help for everybody of us if make patient first, and I think it's God will bless us. I see you in our future webinars. Again, I will be very thankful that you subscribe on my channel. I will be thankful that you will leave your question under my webinars. You can find and ask me personally in LinkedIn profile or in Facebook group. I have created a clinical and pharmacovigilance group. And yes, see you later, and take care, and God bless everybody from us. Bye bye.